Mendelian genetics refers to the patterns of inheritance determined by Gregor Mendel. Mendel is often referred to as the father of genetics because he was the first to determine many of these patterns of inheritance that we commonly use in genetics research. Mendel was an Austrian monk who lived during the 19th century, and he performed hundreds of crosses between pea plants that he carefully controlled. He chose peas that had very obvious characteristics like the color and shape of their seeds and their pods, and through all of these experimental crosses and his careful record keeping, he would log down the types of characteristics that the many generations of crosses would produce. And through these all, he would develop the particulate theory of inheritance. And the particles that he was referring to in his particulate theory of inheritance, we now know as genes. Remember, Mendel had no idea about chromosomes or genes or anything like this at the time he was doing. He simply crossed peas that had certain characteristics and kept careful records of the characteristics in the resulting offspring, and after multiple generations was able to see these patterns. Now, genes, as we've described before, are the regions of DNA that code for particular traits. So these are just sequences of nucleotides and those bases that we've talked about before, the A's, C's, G's, and T's on our DNA. However, one chromosome can contain many different genes. So for instance, let's think up a hypothetical chromosome in a plant that has all of these single genes on it for instructional purposes. So for instance, there's a gene for flower color, or the shape of the seeds, or the color of the seeds, and so on. Now, the location of the gene on the chromosome is called its locus. So what we see here are 4 loci from all these different genes. And these would be on, for instance, a chromosome from the mother or from the egg cell. Now, diploid cells have 2 forms of every gene. Of course, these are 1 on each of the homologous chromosomes. So here we see the same genes on the chromosome from the sperm cell that created this diploid organism. Now, what you notice are each of these letters representing the genes. These are the alleles, the different forms of a gene. And what you can see is that in some cases the alleles are the same, indicated by the uppercase or lowercase nature of the letter, but in some cases they're different. So for example, if we look at this last example, the pod color gene, we see that it has 2 alleles. It has a capital G representing the green color of the pea pod, and it has a lowercase g representing a yellow pea pod color. So these alleles can be the same on both chromosomes, or they can be different, as in this case. Watch the next video where we'll talk about the difference between the upper and the lowercase letters. Now what we want to get to is Mendel's first law in the patterns of inheritance, his law of segregation. And we actually have already talked about this in meiosis, but remember, at the time Mendel had no knowledge of chromosomes or genes at all, yet he was able to determine this simply from the observations he made of the offspring's characteristics. So Mendel's law of segregation starts with the statement that each individual has 2 factors for each trait. And of course, we were just talking about that with the fact that each allele is represented on both the mother and the father chromosome. So we have 2 factors for each of these traits, 2 alleles for each of these traits. And remember that during meiosis 1, these homologous chromosomes separate. And Mendel was able to determine this, and he stated it as the factors, those alleles, separate during the formation of gametes. Unknowingly, he was referring to what happens during meiosis 1. Mendel went on to say that each gamete that's created contains only 1 factor from each pair of factors. And of course we know that after meiosis 2 occurs, where we separate the sister chromatids and we create our 4 gametes from our original cell, we can see how, because the homologous chromosomes were separated, each of these gametes that are created contain only 1 of the 2 original homologous chromosomes, and therefore only 1 of the alleles out of the possible 2. And finally, fertilization gives each new individual 2 factors for each trait. And of course here he's referring to the fact that the one set of chromosomes contained in the egg cell are joined with the one set of chromosomes from the sperm cell, creating the diploid organism that has two alleles for each gene in its nucleus.